So hello everyone, welcome to the HPL seminar this week. It is my great pleasure to introduce our invited speaker, Dr. Lieber. Dr. Lieber is a professor at the Northwestern University with appointments in the Department of Physical Medicine and Rehabilitation, Physiology, and Biomedical Engineering. He received a PhD degree in biophysics with a minor in electrical engineering from the University of California in Davis, where he developed a theory of the light diffraction that was applied to mechanical studies of single muscle fibers. Dr. Lieber's work is characterized by its interdisciplinary nature, an approach that is relevant to those who study biomechanics and orthopedic surgery. He has published over 300 articles in journals ranging from the very basic fundamental science to those more applied ones. He has also pioneered studies of human muscle. Currently, Dr. Lieber and his group have been working on developing state-of-the-art approaches to understanding muscle contractures that result from stroke, cerebral palsy, and spinal cord injury. His lab has been supported by the NIH grant in both investigator-initiated grants as well as a center grant for more than 35 years. Also, in recognition of the clinical impact of his basic science studies, Dr. Lieber has won numerous prestigious awards, including Giovanni Borelli Award from American Society of Biomechanics and Kappa Delta Award from American Academy of Orthopedic Surgeons. Dr. Lieber also achieved his MBA in 2013, and he is also Chief Scientific Officer and Senior Vice President at the Shirley Ryan Ability Lab, which is recognized as the first ever translational research hospital where clinicians, scientists, technologists, and innovators work together in the same space, applying their research in real time to physical medicine and rehabilitation. Aside from his academic and work career, when I asked Dr. Lieber for a fun fact, and he mentions that he only went to the college because he was a failed musician, while his father was a professional musician. I would love to hear about this story more, but before that, we'll be listening his talk entitled as Intraoperative and Laboratory Studies of Human Muscle Contractures. All yours.
Thank you, Sangwon. So I'll go ahead and share my screen. That's okay?
Yes.
Okay. Thanks, everybody. It's nice to be with you. I wish I was doing it in person. I have many fond memories of colleagues in Calgary. Of course, Walter and Benno, and now Art and Rob by proxy, I guess now. But it's great to be with you. I'll be talking today about applied studies of muscle physiology, almost all in humans. And I'd like to start with the acknowledgement of the surgeons who I've been working with, who are most of the source of our muscle. You may know Dave Sutherland was a pioneer in cerebral palsy research, started many of the clinical gait labs in the United States. Jan Frieden I still work with, a hand surgeon now in Knottwil, Switzerland. Hank Chambers is a surgeon in San Diego, California, and Sudarshan Dayanidhi is a physical therapist, PhD at the Shirley Ryan Ability Lab. So I'll talk today mostly about cerebral palsy. I'm going to do it in the context of muscle design, but you know, it's funny. We were just talking about funding. It's really hard to get research funded on cerebral palsy and muscle, because as most people know, cerebral palsy is a brain problem that happens around birth. There's some kind of accident that happens around birth, complications during birth, or excessive birth, etc., length of labor. And when children have cerebral palsy, they end up with movement disorders, and they're classified based on the gross motor function classification score, 2, 3, 4, 5, with 2 being the least severe and 5 being the most severe. Now, I'm a muscle physiologist by training, and I was really not interested in cerebral palsy at all, except I was working in a hospital where surgery right next to us was going on with cerebral palsy, and I'll tell you that story in a second. But let me just give you a background in terms of the way I think about muscle. I think of it as this great composite tissue, and of course, physiologists over the years have done a great job at the nano level and the micro level, understanding, for example, the structure of the myosin filament, the structure and regulation of the actin filament, sarcomere structure, the way sarcomeres are arranged in myofibrils and fibers. And then I guess, yeah, I mean, obviously there's many, many unanswered questions, but as we go from the fiber to the fascicle to the whole muscle, including all the extracellular tissues and cells, it becomes less understood. And yet, in terms of clinical medicine and practice, let's say, of orthopedic surgery or physical medicine and rehabilitation, it's whole muscle properties that are really kind of the things that physicians are treating. I was raised as, I would call it, a sarcomerologist. So here's your classic longitudinal electron micrograph of a muscle, with the sarcomere being the functional unit of contraction of muscle. I say to a first approximation, if you know sarcomere mechanics, you roughly know muscle mechanics, but you can see the Z lines that demark the sarcomere, the A band, which is composed of the myosin filament, other proteins, the I band, which contains mostly the actin filament. And when you measure functional properties, at least of single cells, this is classic Gordon-Huxley and Julian graph, you see this basic relationship that muscle force is a function of sarcomere length. And the brilliance of this study, although I know it's not without some controversy, in my opinion, the brilliance of this study in the 60s was that the sarcomere length tension curve can be defined based on the amount of myofilament overlap at the various sarcomere lengths. So that at very long lengths and very short lengths, muscles develop less than optimal force, and that there's some optimal in frogs, let's say around 2.0 to 2.2 microns. So I was giving my thesis seminar, and I was in a zoology department. As Sung Wang noted, I got a biophysics degree, but I was in zoology. And one of the guys there who studied frog hopping said, why does a frog care if its sarcomeres have this length-dependent property? In other words, what is the normal operating range of a sarcomere in a frog, posed here graphically? Is a frog designed to operate at sarcomere length, or is it long lengths or short lengths, or all across the length tension curve? And I thought that was really a great question. And I'm embarrassed to say I really hadn't thought about it. You know how it is when you're doing your thesis work, you put your head down and you do what you're told, and you do what you're trying to show in your aims. So I did start considering this question and started doing some studies, but I'll fast forward a little bit. I met a hand surgeon when I moved to San Diego, and he was very much interested in surgical transfer of muscles. And so we basically took our lab tool, and we had been using a tool in the laboratory whereby we shine a laser into a muscle, and you know that muscles have the striation pattern. And so if you have a striation pattern along, let's say, the bottom of the screen here, and you shine a laser through it, you get this beautiful diffraction pattern. And the angle of these diffraction orders relates back to the sarcomere spacing. So at least once you have an isolated muscle, you can stretch a muscle and you can measure sarcomere length without damaging the tissue. So because, I mean, it was really opportunistic. It wasn't any great insight, but because Jan was a hand surgeon, Jan Frieden, we basically modified our laser tools to be able to isolate muscles. This happens to be a forearm muscle. By blunt dissection, going in and getting small bundles of fibers, because we're using low power lasers. You can have bigger muscles, you just need more powerful lasers. This is battery powered. We put the device underneath the bundles, shine it through, and when you get these orders shown here on the screen, or you can image them on a photodiode array, you can get sarcomere length as a function of some anatomical measurements. In this case, I'm just showing it being measured with a caliper. But since it's simple geometry, it's pretty easy to get sarcomere lengths once you know the diffraction order spacing. And I gotta say, this is muscle work we did now 20-ish years ago, where we were looking at the wrist extensor muscle. So on the left side here is the wrist flexed. On the right side is the wrist extended. The interesting thing was in this system that with the wrist flexed, the sarcomeres were in the 4 range. With the wrist extended, let's say in the 2.5 range. And if you know your myofilament dimensions in humans, human optimal sarcomere length would be about 2.5 to 2.7 microns. So at least the first, this is obviously very low sample size, one joint movement. It raised the general issue that maybe muscles weren't designed to operate at their optimal length all the time. I got to tell you, when I was teaching physiology, that's mostly what I was teaching students. But this gives me a little bit of pause. And so we started doing a lot of studies of the wrist, prime movers of the wrist, the 2 wrist flexors and the 3 wrist extensors. And I'm just going to boil it down to a length tension curve. It's oversimplified. I'm happy to have you read the papers, and we can certainly have plenty of time to talk about it. But the basic idea turned out to be this, and this is what I would say would be a general rule. I think in general, muscles have a preferred sarcomere length operating range. In general, we don't know what that is, and it's hard to reason it from first principles. What we've done is kind of work backwards. We found that when we measured sarcomere length of the flexors, with the wrist flexed, let's say, the flexors are shorter than the extensors. That's not surprising, because with the wrist flex, the flexor muscles are shorter. Their sarcomere lengths are also shorter. And what we found based on measurement of sarcomere length changes and the moment arms at the wrist, as you go from flexion to extension, the wrist flexors get longer and stronger, and the wrist extensors get shorter and stronger. And so the take-home lesson for us, at least for the wrist, was the wrist joint has obviously flexors and extensors that are antagonistic to each other, but it looks here like the design of the wrist is sort of to have a consistent torque throughout the range of motion and have it be roughly balanced. So we would say the wrist muscle, or let's say the wrist joint, based on its moment arm at the wrist and the muscle sarcomere lengths, is designed to be kind of a stable base, we think, for, let's say, finger manipulation. That's one idea. It certainly is not universal. In fact, of course, as you know, when you look around the body, you see that muscle fiber lengths come in many, many dimensions. I'm just showing you here artistically, taking artistic license, a muscle like biceps brachii with long fibers that run parallel to the muscle, or a muscle like vastus medialis with a bunch of short fibers that run obliquely to the muscle, and then of course everything in between. And if you dissect muscles carefully, as most of you know, there are lots of variabilities, but we classify them into some category, and then we can calculate based on muscle masses and muscle densities. We can calculate, let's say, the total area of muscle fiber in the muscle. And we refer to this as physiological cross-sectional area. It's, to a first approximation, it's muscle volume divided by muscle fiber length. And for example, I'm just showing you one scatter graph here. On the horizontal axis is fiber length of the forearm muscles. On the vertical axis is physiological cross-sectional area. So the vertical axis would be proportional to force production. The horizontal axis would be proportional to length excursion, let's say. And you can see it's kind of a shotgun cloud. Different muscles have different designs. And again, to a first approximation, it looks like some muscles are designed for high excursions. Brachioradialis is a good example. It sort of makes sense because it's got a big elbow flexor moment arm. That kind of makes sense. Some muscles like pronator teres have short fibers, but they have a high physiological cross-sectional area. And if you know where it is, it's designed, it has a pretty small moment arm, so it's designed to produce pretty good pronation moments. And again, like I said, everything in between. So we've measured a lot of these muscles. We've looked at a lot of sarcomere length ranges, and we thought we had this idea for how the wrist is designed. We were essentially doing surgical manipulation of these muscles for spinal cord injured patients to try to reconstruct, for example, let's say you've lost your wrist extensors due to a radial nerve injury or a high cervical injury. Well, sometimes you can take a wrist flexor, like flexor carpi ulnaris, and you can transfer the wrist flex to a digital extensor. But since the digits go down the hand and cross the wrist, it becomes a compound wrist and digital extensor. So we were doing a lot of these kind of models in silica, and we were right next door to kids who were being operated on for cerebral palsy. Oops, sorry, skipping that one. Sorry, here we go. Sorry. These kids had cerebral palsy, and they had fixed wrist flexion contractures. And so since we had measured the normal flexor carpi ulnaris muscle, I say it's normal because all these data are obtained in patients undergoing surgery. So something's not normal. But these patients had nerve injuries to the extensors, so their FCU muscles were normal. They were being transferred into the extensors. But these kids had wrist flexion contractures. And when you went in the operating room, the wrist is flexed, the flexor carpi ulnaris tendon is bowstringing here. And when you cut the tendon, it was obvious that the tendon was retracting. So the muscles are under tremendous amount of tension. And the surgeon next to us said, we really think you guys ought to try to measure these sarcomeres. And I told Jan, well, I'm sure they're just going to be super short. It's not going to be very interesting. We don't even really know what cerebral palsy is, but we better do this because the person asking us to do it is in charge of the operating rooms. We want to keep everybody happy. We didn't have many resources. And the reason I build up like that is I knew what was going to happen. I knew it was a short muscle. I knew it had short sarcomeres. And I say this because when you think you know, and then you go in and find something that's totally different, the totally different thing is probably the truth. In fact, what shocked me was that the sarcomeres in these shortened, contractured muscles it was a short muscle with super long sarcomeres. I mean, super long. If you look at a length tension curve, 3.5 is way beyond optimal. 2.4 is around optimal in humans, but you can see here, the difference in sarcomere lengths would be expected to have profound functional consequences. And we know from measuring children with cerebral palsy that one of the problems they have is they're weaker. And so it's really interesting. They're weak partly because their sarcomeres are long. Now, this raises all kinds of questions, and I'm going to skip a bunch of other studies related to the mechanics of these muscles, but I will tell you that we have done these studies now in flexor carpi ulnaris, soleus muscle, I'll show you some of those data in a second, and then hamstring muscles. And so for the 4 muscle groups for which we have really good sarcomere length data, we have evidence that the sarcomeres are very long, which is strange because when you look at the literature, I would say the general meta-analysis of the CP literature is that the fascicles of kids with CP are slightly shorter than typically developing children. So one of the things we did in conjunction with the Soleus paper, I think to sort that out, was we measured the fascicle length of the soleus muscle in kids about to undergo surgery. So we get a fascicle length. They were either undergoing surgery for a fracture, typically developing kids, or for cerebral palsy. And then in the operating room, we could measure sarcomere length at that same fascicle length. And so we would have a fascicle value from ultrasound, and we'd have a sarcomere length. And of course, if you have the length of the fascicles, and you know the individual length of the sarcomeres, you can calculate serial sarcomere number. And that's the data I'm showing you. So this is kind of the interesting part. And again, this teaches you as a scientist what you learn from your tools. And you can see when we looked at the kids with CP or kids typically developing, this was part of Margie Matthewson's thesis, the fascicle lengths are about the same. And I would say when I look critically at the literature, and Rob Herbert and Lee Barber have done really nice meta-analyses of these in Australia, I'd say on average, people say that CP fascicles are a little shorter, but sometimes they're a little longer like this. But what's interesting is the fascicle lengths in our system were about the same, but the sarcomere lengths were very different. And you can see here that what that means is the CP muscle, while the fascicle lengths are similar, the CP muscle is like this. Similar fascicle lengths, but highly stretched sarcomeres compared to the animal models, which are similar fascicle lengths and let's say normal length sarcomeres. So that leads us to 2 important points. One is a lot of these kids have had contractures for years. You probably know from the mammalian animal literature that when you stretch a mammalian muscle doesn't like to be at an extreme sarcomere length. So if you stretch a rat or a mouse or a dog or a rabbit or a cat muscle, you get long sarcomeres immediately, but then the muscle will synthesize new sarcomeres, add them on, and the sarcomere lengths will become more or less regular. Similarly, if you shorten those muscles, they get short, the sarcomeres get short instantaneously, and they don't like being that short, so they will get rid of sarcomeres and slowly stretch out the sarcomere length and become, again, closer to optimal. So sarcomere number is a highly plastic value. We don't have a huge amount of data on it in humans, as you can imagine, because it's hard to get the data. But the point is, they typically developing muscle will typically seek some optimal value. Now, that optimal value may vary among muscles, but it does change. So the fact that these muscles are stuck at really long sarcomere lengths is weird. And it's actually kind of unprecedented in mammals. And it implies, at least to us at the time, and I still think this, that there was a defect in the muscle's ability to undergo longitudinal growth. Normally, bones have a growth plate and bones grow. As they grow, the muscles which are attached stretch a little. And it's slow, right? It happens on the days, weeks, months timescale. And during growth spurt, it can happen literally on a month. A kid can grow in a month a lot, as you probably know, if you have kids. But as the sarcomeres stretch during growth, just like I told you, that's a stimulus for sarcomerogenesis. You add sarcomeres, they come back to a normal length. What we think is going on with these muscles in kids with CP is there's some defect in their ability to grow. So as the bones stretch the muscles, they don't add the right number of sarcomeres. And slowly, as let's say, you can imagine for the forearm, as the radius and ulna grow, but the muscle doesn't grow with the bones, it slowly gets pulled into flexion. And the reason it gets pulled into flexion, finger flexion and wrist flexion, is that you have a lot more muscle mass on the flexor side of your forearm than the extensor side. So for example, if you just take a cadaver muscle, break the bones and put a frame on it and stretch it, it'll end up like this also, just because of the amount of muscle on the flexor side compared to the extensor side. So you start to look at the literature. And so for example, I like this study that Sylvia Blemker's group published a few years ago in Muscle and Nerve. They just measured the volume of muscles with a bunch of kids with a bunch of different diagnoses. And you can see from this scatter graph, the blue muscles are muscles that are smaller than normal. And the more severe cases of CP had smaller muscles than normal. Now, obviously that could be a disuse kind of thing, but you're starting to build a case, and this is of course what we would do in a grant proposal, starting to build a case like there may be a growth defect in these muscles. And Sudarshan Dayanidhi did a really nice, I thought, meta-analysis where he gathered all the fiber area data in all of the literature in a bunch of kids across a bunch of age ranges. And you can see there's a lot of scatter in the data, of course, because they're different muscles, different even methods. But what he showed, I think pretty convincingly, was that the fiber size of kids with CP grows at a slower rate than the fiber size of typically developing kids. And that started us, so now what's this, mid-2000s, 2010s, this started us thinking about growth in human muscle. And so we did several studies where we started looking at the cellular components in muscle responsible for growth. And he and I wrote a review basically on the satellite cell. And you probably know, many of you know, that the satellite cell is the muscle stem cell that, interestingly, during development, muscles form these giant cells that we talked about, myofibrils turning into whole fibers that can be, tens or 20s of millimeters long and thousands of microns in diameter. These giant cells form by the fusion of single-celled myoblasts. And those myoblasts fuse together, form a big muscle fiber, but not all the myoblasts fuse for reasons nobody knows. It's kind of interesting, right? So you've got these big cells shown here, and then a myoblast just sits on top of it, and that myoblast basically goes into a quiescent state. And you can measure that quiescent state by markers, and the PAX7 transcription factor is one of the markers for a satellite cell, and when its other transcription factors are negative, it's defined as a quiescent satellite cell. So that's what I'm showing you here in this cartoon. These are quiescent satellite cells that are PAX7 positive. They can get activated, and they get activated by a mechanical stimulus, or if you inject a neurotoxin, they can get activated. If you traumatize the muscle, they can get activated. And then they will proliferate and form a new fiber. And then some of them will go back to quiescence. So this process happens all during our lifetime. And satellite cells are responsible for maintaining homeostasis in muscle. They get activated during normal exercise. What's interesting, though, is there are some muscle diseases, for example, Duchenne muscular dystrophy, where the satellite cells just cycle all the time because the muscle is constantly degenerating and regenerating. And finally, these young boys don't have enough satellite cells to make muscle. So we asked a very simple question. What's the satellite cell content of muscle in CP? and this was Lucas Smith's at the end of his thesis, you can see we were able to isolate satellite cells, endothelial cells, inflammatory cells. These are really just positive controls for getting cells out of muscle. And it's a technically sophisticated method, and I'll tell you about that in a second. And you can see here, at least according to flow assisted cell sorting, these muscles have lost like 70% of their satellite cell population. And let's say that's true. If that's true, that's terrible news. And I'm in a department of rehabilitation. What do you do if you're a physical therapist and you're trying to strengthen a muscle or elongate in a muscle and it doesn't have satellite cells? You're going to have huge problems getting that muscle to grow or repair or regenerate or hypertrophy. So therapy, whereas, you know, as a therapist, you're going to apply your tools of overload, functional overload, or stretch or whatever. If the muscle is biologically inactive or biologically compromised, the ability to perform therapy will also have tremendous problems. Now, those of you who are astute and know about journals know that developmental medicine and child neurology is the CP journal, meaning there's a ton of CP stuff in there, but you rarely see cell biology in that journal. And I like to be honest during seminars. The reason is this paper was rejected by many cell biology journals for one important reason. We ourselves had shown that CP muscle is highly fibrotic, and it's well known that when you try to isolate cells from the extracellular matrix of a fibrotic tissue, it's hard to get them out. So the reviewers of the good journals who knew the methods very well would just say, big deal, it's just hard to get these cells out. Like, you need a positive control, like other kinds of cells in the extracellular matrix. And I was really eager to get this in the literature. And I knew that the control experiments were going to take a long time. Like next postdoc, Lucas was going to leave in 2 months. So again, as a scientist trying to be strategic, I go, okay, I know there's technical problems, but I'm just going to publish it in this kind of crummy journal for cell biology. Not a crummy journal, but each journal, you know, has a specialty. Then I started talking to Sudarshan. I said, Sudarshan, we need to become experts in isolating satellite cells, or sorry, identifying satellite cells in intact muscle. Now, obviously, this experiment worked out because I'm telling you the whole story, but basically, think about what we're trying to do here. Here's a section of muscle, maybe 10 or 15 fibers. And in a normal section, you might have 200 fibers. Satellite cells, there's tons of nuclei. That's what the blue dappy stain is. It's literally a stain for DNA. So anywhere there's DNA or a nucleus, you see a blue dot. Notice here that there is one dot with PAX7. Remember I mentioned that's the transcription factor. That tells me this cell that's expressing this transcription factor is going to be a muscle cell, but it's not one yet. It's this green dot right here, blown up right here. Now it overlays this dot, so it's a green dot overlaying a nucleus, and if you have the laminin stain, which is shown here in red, it's inside the basal lamina. So remember, a satellite cell sits on top of the sarcolemma, underneath the basal lamina, and is a nucleus itself, has a nucleus. So those three criteria define a satellite cell. So what you do is you hire an army of undergraduate students who love to learn how to cut tissue and stain it, and you have a cheerleader like Sudarshan, and you basically say, we're going to count satellite cells in CP muscle, normal muscle for the next year. And it took us a full year to do this. And when we did it, the good news is the data are shown, let's see, down here. This is number of satellite cells per 100 myofibers. You can see in CP, that number is significantly low. And this is the FACS data I just showed you, whether by cell sorting or immunohistochemistry. I think it's a real result. In fact, I think this is probably one of the most important findings for us to try to understand the etiology of the disease of contracture and what should we do to fix a contracture. And about this time, I was starting to study, I mean, I'm a physiologist, I'm not really a cell biologist. I was starting to study stem cell biology, especially in muscle. It's a big topic, so there's a lot of literature, there's a lot of really good muscle people in that area. But I realized I was out of my league, and I hired a real live cell biologist, a guy named Andy Domenighetti, who's a Swiss guy who got his PhD in Melbourne and was a postdoc at the time in San Diego. And we basically started growing satellite cells from human muscle. And it's kind of complicated. We were going to start a company to inject satellite cells into kids with CP to try to get rid of their contractures, sort of like Helen Blau did at Stanford when she tried to inject satellite cells into boys with Duchenne muscular dystrophy to fix dystrophy. So it was the same idea. And whenever you have a business, you always have milestones. The first milestone was, can you grow muscle? So I remember I actually entered a contest. I won. I got some money. And the reviewers, like a Shark Tank, they said, what's your first milestone? I said, my first milestone is take muscle biopsies from kids with CP, grow muscle. They said, how long is that going to take? I said, it's going to take me 2 months. Because we were growing cells from mice, so who cares, mouse, human, once you're at the cellular level, who cares? So to my chagrin and to our company's failure, we couldn't make a muscle. Isn't this interesting? So now I'm showing you, this is a picture from cell culture. So on the left-hand side is typically developing satellite cells isolated using FACS sorting versus cerebral palsy satellite cells isolated by FACS sorting. The red immunostain is slow myosin heavy chain. The blue is that DAPI, like I showed you before. So here you can see an example of a myotube. This is going to be a myofiber when it grows up. It's a multinucleated myotube expressing slow myosin heavy chain. And if you looked at it, let's say with phase microscopy, you'd be able to see baby sarcomeres. Okay, so this guy's on his way to being a muscle fiber. But look over here. You can see a little bit of fusion in these myofibers. First of all, there's a whole lot more of the connective tissue junk around there. This is a wheat germ agglutinin stain, which stains things like collagen and extracellular matrix proteins. But when you see a lot of this green junk, it means that the extracellular matrix is there, but the cells just aren't happy to grow on it. And we did a bunch of assays and showed essentially that these muscle cells, the satellite cells, can proliferate and divide, but they just don't continue down the developmental pathway. And there are lots of proteins you can measure, but if you are, oh yeah, I was going to say, the interesting thing is, and the important conclusion is, CP, we know, is not a simple genetic disease like a primary myopathy like Duchenne muscular dystrophy. Duchenne muscular dystrophy is a mutation in the dystrophin gene resulting in destabilization of the myofiber and all the problems associated with it. CP has a few genetic predispositions, but it's not a simple point mutation or a genetic problem in a protein. So the fact that the satellite cells, which presumably were developing normally until the CP event happened, the fact that they were altered implies some other mechanism of disease, an epigenetic mechanism, a mechanism on top of the genetic profile of the kids. And so we started measuring all the kinds of things. This is where Andy's genius comes in. Andy started measuring the kinds of things you measure that are epigenetically regulated. So for example, it turns out, and again, I'm not the cell biologist, he is, but one of the most common ways to change a gene's expression level is to methylate it. So when a gene is hypermethylated, it decreases the expression. It decreases the amount of that protein. So the integrin beta 1 protein is a protein on the cell surface that allows a cell to grab the substrate. When a cell can grab a substrate, it will develop. If it can't grab, it won't develop. So Andy made a lot of educated guesses at proteins like this. I can show you other examples. But what he showed was this protein, this gene is hypermethylated. And when you look at the fusion index, that is the amount of fibers that are actually fusing together, fractions of fibers, that actually goes down dramatically. And so his conclusion was hypermethylation of the integrin and other genes, other related genes, causes a hyperproliferation of the tissue and lack of fusion. So he started thinking of this, as a good biologist would, as like a hyperproliferation disease. So you think, okay, what other kinds of diseases result in hyperproliferation? And obviously the most common answer to that is cancer. And the good news for us is that there's a tremendous number of FDA-approved drugs on the market that are designed to demethylate genes in cancer, especially blood cancers in children. So here is Vidaza, which at the time was made by Celgene. It's a demethylating agent. So Andy just said, okay, step one, let's just try demethylating. Try to get rid of these methyl groups and see what happens. And he did. He demethylated the genes with azacitidine, is what's abbreviated here, you can see the amount of methylation dropped and the amount of fusion increased. In fact, on average, in this paper, Andy showed that he could rescue, we call it rescuing the phenotype of the CP satellite cells by demethylating the promoter and allowing development to proceed normally. And I'll tell you, we're in the middle of a clinical trial where we are trying to figure out, because the nice thing about using a drug like azacitidine is it's been on the market for 25 years. For those of you who are muscle nerds, azacitidine was actually used in the 70s to discover MyoD. MyoD is a classic transcription factor in muscle that acts by demethylation. So azacitidine had been used forever in muscle. It's probably got 25 years on the market. In fact, it's been on the market so long, it's essentially generic, and these companies are now having to come up with new formulations. But what we're doing now is we're trying to figure out what's the right dose in children to give them azacitidine and try to work out these contractures. Because as I told you before, if you're trying to do therapy and make these muscles stronger, they have to be able to grow or elongate. And if they're biologically, let's say, incapacitated, that therapy is going to be really hard. But our idea is, okay, well, let's turn them back on biologically, and then let's apply these splinting methods or these stretching methods or these exercise methods. And I tell Andy, I know we're on the right track because so far none of our grants have gotten funded. But we've been able to do some of these studies with philanthropy and that kind of stuff. And I hope to be able to report in the short term that we're able to do some things in kids with contracture. And I'm happy to talk more about that during our discussion, but I did want to bring up a couple of other things. And that's just sort of the whole idea of satellite cells being the causative problem, because maybe satellite cells are an epiphenomenon. They're just going along for the ride. So this is work that I'm now telling you that Sudarshan did, and this is becoming his area, where he has now trying to change satellite cell number experimentally. And so remember, I've showed you both of these graphs before. I showed you one graph where satellite cell number was down in CP muscle. And I also showed you that CP muscle has less sarcomeres in series. And I implied that cause and effect. Decreased satellite cell number causes decreased sarcomere number. I implied it, and I even said it. But is it true? I mean, maybe they just go together. Maybe that's, another epiphenomenon. So these are data from humans, obviously on both sides, but we wanted to try to manipulate the system. And so we went to a colleague who is a satellite cell expert, and Charlotte Peterson at the University of Kentucky developed an inducible mouse model which kills satellite cells. And that's shown in this slide. So now I'm telling you about mouse muscle. Everything else pretty much I've said has been in humans. Now this is a mouse. So we have wild type mice, and we have this special strain that Charlotte and her colleagues developed, where it's a PAX7 promoter. Remember I said PAX7 is a transcription factor for a satellite cell. So PAX7 will identify all satellite cells. And then they genetically modified the genome of a mouse to put diphtheria toxin downstream of PAX7. And to make it even cooler, they made it so it's inducible by this drug, tamoxifen. Tamoxifen is an estrogen analog. So if you engineer the mice right, and this is very common now, again, I don't know how much you all deal with these models, but it's very common. You can basically feed the animals tamoxifen that will turn on this transgene that will express diphtheria toxin. And in an adult animal, like let them grow up, be whoever they want to be, go to college, have families, have friends, send their kids to college, send their kids to school. But then when you feed them tamoxifen, they will kill their satellite cells. Okay, so here's the experiment. This is work Matt Kinney did. Matt Kinney is an orthopedic surgery resident who was a mouse doctor for a year in the lab. So what you do is you feed the animals tamoxifen for 5 days, all the animals. Then what we're going to do is we're going to take animals, wild type and transgenic. We're going to immobilize the hind limb in in dorsiflexion. So if you dorsiflex the mouse ankle, you stretch the soleus muscle. That's the easiest one to make sarcomeres out of. It works in other muscles too, but soleus gives you the best signal. So the idea here is if you dorsiflex one side, you'll add sarcomeres, leave the other side uncasted, that'll be your normal, and then you harvest them and analyze them. Now, your wild type mice ought to have an increased sarcomere number. And if your hypothesis is that losing satellite cells is going to cause a loss in your ability to make sarcomere number, these guys won't increase their sarcomere number, right? Because you got rid of a lot of their satellite cells here. So here's the result. And interestingly, both groups made roughly the same number of serial sarcomeres. So, here's the tamoxifen treated group, the transgenic group. The vehicle treated group is over here. And you can see that even with 30% of your satellite cells, we didn't kill all the satellite cells. That's part of the problem with the model. It doesn't kill all the satellite cells. But even though we didn't kill all the satellite cells, we still got a significant increase in serial sarcomere number, pretty much the same as vehicle treated, transgenic or wild type mice. I'm not showing you the wild type data. So that was a little disappointing because we thought we would have a big effect by getting rid of the satellite cells. But here's the interesting part. The quality of the muscle here, even though it had the right number of sarcomeres in series, the tissue wasn't in good shape. So here is H&E section up on top of vehicle treated compared to tamoxifen treated transgenic. And look what happens. The vehicle treated definitely add new myofibers at the end of the fibers. They're centrally located nuclei, meaning these are regenerated muscle fibers. The similar thing happens in terms of there are some fibers that get regenerated in the tamoxifen treated, but there's all kinds of other connective tissue. So this the muscle in the tissue that doesn't have the right number of satellite cells isn't the right quality, and it certainly isn't the right strength. You can see that when you measure the area fraction of muscle fibers. So this is actually kind of promising because it turns out kids with cerebral palsy have a lot more fibrous tissue in their muscle cells than typically developing kids. So we thought an even cooler experiment would be not so much see if they can add sarcomeres without satellite cells, but see if we can actually give the mouse a contracture. And that's a different kind of experiment. Same system. So we have one group of animals that have the inducible promoter that will kill satellite cells, and one group that doesn't, is vehicle treated, so the satellite cell number is normal. On the right side, now for 2 weeks, we're going to start with the ankle in plantar flexion. So now we plantar flex the ankle, we shorten the soleus muscle, so we get rid of soleus sarcomeres. Because remember I told you before, if sarcomeres get short, they don't like that. So they're going to get rid of sarcomeres and bring them back out to length. And you can imagine the sarcomere number will decrease. And when the animal is out of his cast, he or she will have a plantar flexion contracture. And then they work it out. So here's the experiment. For 2 weeks, plantar flex them, get rid of those sarcomeres, give them a plantar flexion contracture here, and then keep them in the cast, treat them with tamoxifen. Now we get rid of the satellite cells, and now we take off the cast over here. So here's an experiment where after a month, you take the cast off an animal that was given a plantar flexion contracture, but we got rid of his satellite cells after the contracture. And now we're going to see if he can work it out. And the interesting thing is, at least over a few weeks, he doesn't, or she doesn't. So the green lines are the ankle angle of the animals that were treated with vehicle, and the plantar flexion contractures are shown with the animals who were treated with tamoxifen. And when you measure serial sarcomere number, they didn't really recover. And so this is a proof of concept experiment. It certainly doesn't prove anything in humans, and which is a very hard experiment, you can imagine. But it does show, at least conceptually, that if you get rid of satellite cells in mammalian muscle, happens to be a mouse, and have them try to add sarcomeres, you have trouble doing it. And the sarcomeres that you do add are of let's say, of a lower quality. So this at least is consistent with the idea that I presented, namely that in contracture, because of an epigenetic mechanism, which we haven't talked about at all, you hypermethylate the promoter of the myogenic genes. There's a zillion of them. By hypermethylating them, you prevent their expression and therefore favor satellite cell proliferation over differentiation. And that is what we think results in the contracture. So if I were to summarize these data, I would say, first of all, I think contracture is real. I know it's complex. I think it's uniquely human. When I read grants, I don't think most of the grants that I read in mouse, cat, rat, and rabbit are really a contracture. There are ways to make muscles stiff and makes joints stiff, but I think contracture is way more complicated. We can talk about that. In the muscle, you can tell I mean, I'm a muscle physiologist by training. Shouldn't I have stimulated a muscle or activated fibers? Yeah, but the point is, they're all pretty normal. So the passive muscle mechanical properties are altered to a much greater extent than the active mechanical properties. And I say it's bad news. And the reason it's bad news is that we know a whole lot more about active muscle contraction than passive mechanical properties. And that's an area of study. CP muscles have trouble growing. I think it's because of the lack of satellite cell number and quality. And therapy, which comes in lots of flavors, is hampered because we lack knowledge in several specific areas. And especially, I would say, the whole thing about how the extracellular matrix is put together, how it's regulated. Of course, satellite cell development and function is a big area of study, yet to be defined. And I said this, actually, we had a meeting that Walter was sponsoring up in Canmore, where I said, we really have trouble because we need some new tools if we're going to study humans. So you think about the corner I'm backing myself into. If I think that human muscle contracture is a uniquely human problem, I'm committing myself to studying humans, which is why I moved to Chicago. But I need some new tools to study humans. So one of the tools we're working on is to acquire human data is measuring sarcomere length in humans with basically fiber optic probes that we can slide into muscles. This is work I did in San Diego in collaboration with Stoyan Radic, who's a photonics expert, where basically you put a zillion wavelengths into muscle, the sarcomeres reflect them back, and if you do a zillion wavelengths, you get an unbelievable signal to noise ratios. And so from these data, you can get passive and active sarcomere lengths from contracting muscles. The trick is poking fibers in the muscle, and of course, this muscle is kind of fibrotic, so that's a little bit of a problem. I told you already that extracellular matrix is kind of messed up in human muscle. Normally, this is work from Lucas's thesis. Here's a cross-section of CP muscle. Here's a cross-section of typically developing muscle. We tried to make it a fair comparison by adjusting the laser intensities on the confocal, but you can see there's a lot more stuff on cross-section and longitudinally. But what can if you're an engineer and you're trying to do a structural model, what can you do with this? These models, these images are just too low resolution. So we have been trying to come up with ways to structurally image the extracellular matrix. And here on the left-hand side, you can see serial sections imaged by electron microscopy, and you can see all this stuff out in the extracellular matrix. Those are cables of collagen. And we've generated a huge amount of data in terms of collagen content, cellular content, spatial location, but it's really hard to analyze. So we're trying to develop tools to analyze these 3D images and then try to come up with these structure-function relationships. And that's sort of where we are. A lot of studies to do in the future, but I'd be happy to talk with you about anything ongoing and where I think the future lies in this area, and let you watch my beautiful movie. That movie took so long to get. I've only got really good movies, because think about this. These are 70 nanometer sections that cover about 10 millimeters of space. So that's tens of thousands of sections. And of course, you can see as you're watching the image, the sections drift. So we get maybe, if we're lucky, we get several millimeters of very high resolution data, but it's probably not enough. And so again, we're looking for new tools. By the way, way up here in the corner, that's a fibroblast with all this little tentacles and all the little sections it goes through. And that's this blue cell right here. And here you can see coming off to the section, that's a little piece of tendon. And of course, it's packed with collagen. So we watch these movies like most people watch Netflix, and they go on forever. But I'm happy to go ahead and take questions, and I'll stop there. Thank you.
Thank you very much, Dr. Lieber. I really enjoyed your talk. So if you could now unshare your screen. Thank you.
Yep.
So I also would like to kindly encourage everyone to turn on your camera if you feel comfortable with this, so we could see each other. I think then we can have a more active discussion period. Thanks for doing that. So if you have any questions for Dr. Lieber, please use the icon raise hand button, which can be found if you go click reaction icon at the bottom of your Zoom screen. Also, if your Zoom version does not show you that the button there, then please also feel free to send me a Zoom message via Zoom chat, and I'll call on you when it's your turn. Now the floor is open for the question. Actually, let me start asking the question.
Sure.
So about the satellites, the number of the satellites, and then the growing function. So I'm just curious about, would there be any mechanisms or any other factors that could contribute to like killing the satellite cells? Like, so I'm just curious what determines the lower number of the satellite cells.
Yeah, the real answer is we don't know, but there are several possibilities. I think the most likely one is related to the Duchenne muscular dystrophy question. It's pretty well established. I mean, satellite cells have a finite number of times they can replicate, and over a 70-year lifetime, even in a healthy 70-year-old, the number of satellite cells decreases just because they can only replenish their own stem cell niche so many times. So I think the most likely mechanism of the loss of satellite cells is hyperproliferation and essentially senescence. So the way it's, cells have kind of a built-in counter. And you can measure, for example, the length of the telomeres that are involved in mitosis in the cells. And the telomeres get shorter and shorter and shorter, sort of like God snips off one little length of telomere for every division, and they get shorter over time. Telomere length in Duchenne muscular dystrophy changes dramatically over the child's lifetime, which is shortened. Right now, we have a fair amount of telomere data in the kids, but our youngest kid is probably 3 years and some months, and our oldest is, we have adult people with CP. So far, it's not obvious that over, let's say, the 25 years of time we have, that's really changing much. So that's kind of bad news, because that would have been kind of cool if a kid is born, what's their satellite cell number, what's the prognosis, could you predict treatments and function and even prevention perhaps. But our youngest kid also has decreased satellite cell number. So our current thinking, and we have no support for this, is that they're born that way. So by the time the muscles develop in utero, and of course we don't have any sub-year data, but that they're born with a decreased satellite cell number. And if that's true, that sort of becomes the bad news. Now, and of course, and then the question is, how did that happen in utero? We can talk about that.
Thank you. I see, Benno, you have a question for Dr. Lieber?
Yeah, thank you, Rick. It's fascinating. Like a criminal story.
It's crazy, isn't it?
Yeah. When you do surgery, or your friend does surgery at the hand, what do you expect? Why do they do surgery, and what do they change?
So that's a great question. The typical surgery, and it sort of makes sense. I think it does make sense from a biomechanical point of view. So you've got a wrist flexed, and the sarcomeres are stretched out. I think the surgeons basically give up on the muscle. They say, okay, that muscle is not going to grow, but what I'm going to do is I'm going to cut the tendon, straighten out the wrist and fingers, and now the muscle has cut the tendon so the muscle retract, and you either insert a graft or do the kind of surgery where you can kind of lengthen the insertion. So they put the muscle back at a new resting sarcomere length, which is usually pretty appropriate, and then retrain the muscle there. So the good news is you've relieved the contracture. You probably have made that muscle more functional. The bad news is you haven't changed anything about the muscle, so you're probably going to have a recurring contracture anyway. But the idea is maybe you can buy some time, or maybe you can get the child over their growth spurt, do the surgery, and kind of biomechanically correct. Obviously, you wouldn't go back to a normal situation, but you would definitely be more functional if you could open up the hand and move the wrist.
And it works?
Works pretty well. I mean, it's surprisingly effective. Once the surgeons understand, it's kind of funny. They call it, I go in and I do a tendon lengthening. And I say, wow, okay, I'm a muscle guy. You're doing a muscle shortening. And they go, yeah, that's what I mean. So once you teach surgeons the biomechanics of a tendon lengthening, and you have to teach it that way, they get it and they do a pretty good job. And they all say, oh yeah, give me a laser, I'll go ahead and measure it. And then, Jan and I at one point thought everybody in the world was going to start measuring sarcomeres with lasers. And it turns out it's tricky. And Jan is an extremely good muscle surgeon. So he's really good at blunt dissection. And most surgeons aren't. So basically we give dimensional guidelines to surgeons. If the forearm is this long, we've literally come up with a chart. You look up in a chart, here's how long the forearm is, here's measure of the origin of the muscle to the muscle tendon junction, here's how long you should make your gap in your tendon lengthening. So it ends up being sort of like an erector set to play with.
Thank you.
Yeah.
I see, Eric, you have a question for Dr. Lieber?
Yes, thank you. Fantastic presentation. I really enjoyed it today. My question is, how uniform are contractures in CP, patients?
You mean across the body or across muscles?
Across the body.
Well, so CP comes in several flavors, right? You can have CP that affects all your limbs and both sides of the brain. You can have CP that just affects one side of the brain. So you could have a hemiplegic CP with a perfectly functional arm on one side and a dysfunctional arm on the other side. But let's just take I think the simplest example is like a diplegic, where both sides of the body are plegic. The muscles, it's very interesting. This is part of the mystery. Like Benno said, it's like a mystery novel. Wrist flexors, finger flexors, elbow flexors are highly affected. So wrist flexion and elbow flexion, wrist flexion, finger flexion, and shoulder internal rotation are kind of the ultimate contracture posture. Okay. Knee flexion is another one, and so is plantar flexion. But you never see a kid in CP with a knee extension contracture or a dorsiflexion contracture. And no one really knows why. It is interesting that those tend to be they tend to be very strong muscle groups, hamstrings versus quads, plantar flexors versus dorsiflexors, finger flexors and wrist flexors. Obviously you have two sets of finger flexors, only one set of finger extensors. They tend to be the stronger joints, but I think there's something kind of biological or neurophysiological in there. I'm not really a brain guy. I'm working more, obviously at Ability Lab, I'm working more and more with brain people, but I have a feeling a lot of this has to do with sort of normal input that goes into the muscle from the central nervous system, and then how it gets disrupted. Because the $100,000 question is, what's the change in the brain that causes the epigenetic modification of muscle? And I think the answer is, this is going to be crazy, but I think it's brain inflammation and subsequent change in growth factors that are the muscles are bathing in. I think that's what's going on. But anyway.
And then a quick follow-up with that. With these muscles that do not have contractures, are they, say, lacking these healthy number of satellite cells or other markers or indicators of healthy muscle function as well, or are they?
Yeah, I only have one data point, and that is because you can imagine you're talking to parents, we're operating on your child, and we say, hey, we're going to take a little piece of your hamstring muscle. It's not going to help you, but it could help other people later. Oh, and by the way, would it be okay to take a little piece of quadriceps? And the answer is almost always no. In fact, I've stopped asking. So I can't answer that question. The good news is there are some situations, especially in the upper extremity, where you've got more exposure. So we've got one paper where we've looked at wrist flexors and wrist extensors. And actually, it's kind of interesting. So it's a wrist and finger flexion contracture. We took a little bit of FCU and a little bit of ECRB, and we just compared them histologically, okay? Like you would if you were a pathologist. And it turned out the pathology of the wrist extensor was greater than the pathology of the wrist flexor. So this is why I'm thinking, okay, there's a systemic body-wide change in muscles But the actual posture of the child will end up being determined by, how big their muscles are on which side of the joint, not some intrinsic difference between, let's say, the biology of the muscles. But I'd say that's a very soft comment. I wouldn't, if I'm wrong, I won't be surprised.
Awesome, thank you.
Sure.
Yes, Walter, please go ahead.
Thanks, Rick. Always a pleasure, and I always learn something new every time I listen to you. And
Even though you've heard the same story about 4 times.
yeah, but and I'm asking the same question.
Okay, go ahead.
I'm asking the same question that I asked in 2019, and that is, some muscles in the CP children, as you just mentioned, are spastic, and then a lot of the other muscles in some CP patients, at least, are completely normal. They grow normally, and one leg is absolutely normal, or one arm is absolutely normal. And do you know what the satellite cell number is in the normally developing muscles of CP children? Because I've just been interested, if that happens systemically over all muscles, and some then develop normally and others do not, or do the normally developing muscles in CP children have a normal number of satellite cells, and only the spastic ones have the reduced number that you observe?
It's a great question. I just can't answer the question. I don't know. It's so funny because I don't know what's going on, but I would say 20 years ago, parents said yes to everything. I mean, you wouldn't believe what we did 20 years ago.
Yeah.
It was ethical, but it was more invasive. And I would say, and I don't think it's Chicago, because I'm working still in Chicago and San Diego. Parents are much more hesitant. And trust me, I'm a dad and a granddad. I love my kids. I'm not sure what I would do if I were the parent, but we've asked so many families for I always say it's an eraser-sized piece of muscle from their quadriceps. And we have 0 data so far. That's another reason, like even our laser, the good news about our laser is it's pretty good at figuring out nuclear number, because DNA is very optically active. So nuclear number we might be able to get in normal muscle. And then we'd have to do a little bit of calculations and things like that. But I can't answer the question. Sorry.
The other thing, and as you know, in an animal model, we can activate a muscle artificially. We put a nerve cuff electrode in, we can activate it, or we can also block it. We can block nerve signals. And so many years ago, at one point, I thought, what if we could recognize in a spastic muscle that has contractures, what if we could recognize the real signal coming from the brain that you actually want to have, and signals that you don't want to have, and we're able to block them? And let's assume that actually that would be possible. Do you think that the muscle might develop normally then, so that really it's the signal from the wrong signals that comes through the nerve that actually then produce the phenotype that we see in spasticity?
I actually do think that, yes. Can I show you one figure from a paper that just got rejected, so I know it's pretty good? Hang on.
I'm sure Song 1 will allow you to do that.
Okay, I'm gonna share this screen again. Sorry, I know this is probably a slightly awkward, but that's okay. Yeah, here we go. So you're seeing this figure on the left-hand side, a scatter graph, on the right side, a bar graph.
Yeah,
so this is an opportunistic study where we had a bunch of muscle in the freezer, and we had the course of treatment, and look, it's really a crazy result, which is why I'm having so much trouble. As I said, I don't have a huge amount of data on the patients. They were San Diego patients who I can't find now, but kids of different ages, all of whom were having surgery for a variety of things in CP, mostly releases here and there, all upper extremity, and different ages. And the kids who had Botox actually ended up with bigger muscle fibers in their section. So this is histology from the muscle. And if you look on average, the kids who had been treated with Botox had larger fibers. Now, these are still smaller than normal. Normal muscle fibers in these kids are in the, let's say, in the 60 micron diameter range. So I wrote in the paper almost exactly what you said, Walter. I said, I think what's happening is the central nervous system is bringing in bad information, and it's causing not just, I can't activate my muscle, but it's causing atrophy, active processes, or something like that. And in this case, if you block the neuromuscular junction with Botox or other things, you allow the muscle to grow. and you know, this experiment is complicated for a bunch of reasons, but I think exactly what you're saying is very possible. And I think you're getting to the point which I thoroughly agree with, which I think is the neuromuscular junction is way more than just action potentials. And you guys know this because, right, when you denervate a muscle, it's more, it atrophies way more than simply losing fiber size and muscle mass. It actively degrades itself. So the neuromuscular junction being intact is very important to a muscle. And I would love to have someone say, we need a all-out blitz on the neuromuscular junction to do what you said. Get rid of the bad signals, let the good ones through, and hopefully help with rehab.
Great. Yeah. Thanks.
Isn't that crazy?
Yeah. Yeah. It is interesting.
Yeah. Yeah.
It is interesting. I have another question, but I think I defer it to the end if you have a little bit of time, because
Sure,
I'm really interested. I'm really interested in this in vivo time-dependent sarcomere length measurement system that you
Yes.
say. But I see there's other people wanting to ask questions.
Okay.
So maybe at the end, if you have time, we can briefly talk about.
Of course. Happy to. Yeah. Sure.
I see Art, you have a question.
Sure. Hi, Rick. I hope
Hey.
you don't mind if I ask some just incredibly naive questions.
Oh, I thought it was going to be a math problem, Art, and I was getting nervous.
No, that's nothing like that. So I'm just curious how the satellite cells know to do their thing. So like mechanically, is it a mechanical signal, first of all, or is it all you need is damage? And then you were also talking about muscle becoming more fibrous, and I'm wondering what triggers that to happen.
Mm-hmm. So there's pretty good data in the satellite cell literature. So I told you, satellite cell, it's its own cell, so it's outside the muscle membrane, but it's underneath the basal lamina. So it turns out there's dozens, if not more, proteins that cause this sandwich to inhibit growth and proliferation. In fact, satellite cells, the other cool thing about them is they consume very little oxygen. I mean, they really are almost in suspended animation, quiescent. For example, I'm getting muscle from Austria these days. It takes about 2 days to get it from Vienna to Chicago. The only thing that lives in the muscle that they send me is the satellite cells. All the other tissue dies. Isn't that interesting? So there's contact inhibition, and when you release that, and you can basically release it chemically or mechanically, when you release it, it's a signal to grow. And you can do those experiments in culture. The idea is that so long as everybody's happy and the satellite cells in its normal niche, they call it, it will be in suspended animation. But when a mechanical damage happens or a physical shearing, when the shearing gets high, you turn them on. So I would say it's mostly mechanical, because even the chemical effects are indirectly mechanical.
Okay, and then how about when muscle becomes fibrous? Is it a similar
okay, that's
thing
another
to
interesting
build?
thing. So all I talked about was satellite cells. Satellite cells are extracellular. But if you look in that connective tissue schmutz that I showed you, there's lots of cell types. There's fibroblasts, right? There's other types of stem cells, so-called fibroadipogenic progenitors, cells that can become fat or they can become connective tissue. And there's a few other minor players. Those cell populations exist in equilibrium, and there's a competition or something between them. If you get rid of any one cell type, the others take off. It's like the police department, right? So long as the police are balanced with the criminals, there's a homeostasis. As soon as you get rid of satellite cells experimentally, the fibroblasts go nuts. And I think the majority of the collagen that's laid down is by fibroblasts. In our mouse model, the majority it's not the fibroblasts, it's this other cell type that we call them FAPs, fibroadipogenic progenitors. For some reason in mouse, FAPs go nuts when you get rid of satellite cells. I'm not sure that expands to human, but I would say normal ECM amount is a result of normal synthesis and degradation by all the right cell types and all the right combinations. If you get rid of one, same thing happens in Duchenne muscular dystrophy. If you get rid of one, usually fibroblasts take over and just start proliferating and making collagen like crazy.
Well, I think that's really interesting that there's kind of a competition going on, because one thing I've always wondered about is what's the difference between a contracture and just sort of holding posture? Because I've heard that, let's say, pork shoulder is a muscle that is, to me, it sounds like it's kind of like in a contracture condition most of the time. It's supporting body weight and it doesn't shorten that much. If the pig is just standing around, and then the response to that is it has a lot of collagen, and then that's why you're supposed to cook it differently. But then, so what's the difference between that and a contracture where, are the conditions, is there something about the conditions that are very different, or could it be kind of similar conditions, but maybe because of a lack of satellite cells that you get more fibroblasts going on?
Yeah, that it's a good question. I can't fully answer, but I can tell you that at least in musculoskeletal medicine, there are contractures that are simply a result of the peripheral muscle being shortened chronically. So for example, if you break your forearm as a kid and your radius and ulna shorten, your muscles will come to a new equilibrium. They will shorten as well. And if you try to elongate that bone, you're going to have to do it over time because the muscle has chronically shortened. And some people refer to that as a contracture. Or you can actually, you can dislocate a joint and then it doesn't rotate anymore. Like it's very common in shoulder injury patients, rotator cuff injury patients, where they stop using their shoulder and the muscles just shorten and the joint contracture happens. The kind of contractures I'm talking about are, they go along with an upper motor neuron lesion, so a spinal cord injury or a head injury of some kind, a brain injury of some kind. And that's where, like we were just talking with Walter, that's where I think something is going on where the peripheral nervous system is now sending new signals to the muscle, or the normal regulatory mechanism for muscle mass, which is brain, pituitary gland, adrenal glands, cortisol, all those things that are normal thyroid hormone, growth hormone, et cetera, those things get upset. I don't think we really know how any of that affects something like a satellite cell in a muscle. So I have a new junior colleague who actually is a cancer biologist, because in cancer, you get a pretty well-defined mess up of this hypothalamic pituitary adrenal gland axis and concomitant muscle wasting beyond what you would get just by the inactivity associated with cancer. So I think I think all that stuff is yet to be sorted out. And I think it will turn out that there will be a bunch of different flavors of contraction. They may look the same, but I think they'll have different etiologies.
Great, thanks.
Sure.
Rob, please go ahead.
Hi, Rick. The skeletal muscles on the arm, it's unfortunate, it's unpleasant, but the skeletal muscle, the costal diaphragm is critical to life. And so if you were to lose sarcomeres in series, the outcome would be a reduced vital capacity. I'm wondering several possibilities. Have people looked at the diaphragm in these children? Do they have a reduced vital capacity? And by what you're showing on some of the arms, it looks so dramatic. I would suspect if you did that to a costal diaphragm, you would die. So do CP children die from respiratory failure very early because you can't do it later because you're not going to live that long? Or is in fact the diaphragm muscle protected and not suffering from this issue?
So
Any
great
thoughts?
question. Yeah, we have a guy in our hospital who does diaphragm ultrasound. So first of all, CP kids do have breathing. They need respiratory therapists. They have breathing issues. They have to exercise breathing a lot. Now, it's not like a kid with Duchenne muscular dystrophy. They will literally die because they can't breathe because their diaphragm muscle degenerates. But these kids do have fibrotic well, I'll say they have diaphragms that have more echogenic ultrasound signals, and they do have decreased vital capacity. I actually hadn't thought much about the sarcomeres in there, but I wouldn't be surprised if there's some shortening, but it's not, I would say in general, it's not life-threatening. This life expectancy for a child in North America with cerebral palsy is a normal life. So it's not, maybe I'm saying it's not progressive as you might expect, but it's definitely decreased performance. There's no question.
Yes, because I mean, the whole purpose, the method that the costal diaphragm works is the long range in which it operates over. It doesn't have to generate a lot of, well, it does generate a lot of force, I guess, but it's a huge range. So if you cut out sarcomeres, you just direct impact on vital capacity. But on a related
good point, that's a really good point.
What about cardiac muscle?
I can't say I know any studies. I mean, again, in the clinic, you don't hear, you get a lot of referrals to pulmonologists from the CP clinic. You don't get many referrals to cardiologists. That's a good question too. I don't think I have enough data, but it's not the kind of thing I've heard. I mean, I'm no CP expert, but I haven't heard much about that.
What about smooth muscle? Are the satellite cells for smooth muscle, cardiac muscle, skeletal muscle, are they similar? Are they all affected?
They're actually different. Smooth muscle and cardiac muscle have very different abilities to regenerate. Cardiac muscle actually has almost no satellite cells. So it's kind of funny, but you also, cardiac hypertrophy is actually a disease, right? If your heart gets bigger, you have some kind of heart disease. And almost always that disease is associated with fibrosis and things like that. Smooth muscle, I don't know about. I haven't thought about it. These are great questions. I haven't thought about satellite cells and smooth muscle. I don't think I know the answer. Maybe someone else does.
So with the diaphragm, I mean, if you're not going to get a piece of quadriceps, you're not going to get a piece of a diaphragm.
Yeah.
But is it? I mean, my thought was that it's the critical muscle that you can't do without.
Right.
And I was thinking, is there some level of protection? Is there something that, if they're not dying, then...
Yeah. Well, I wouldn't be surprised. Let me put it this way. As you know, the diaphragm is very slow, highly oxidative. Highly oxidative muscles have lots more satellite cells than less oxidative muscles. So it's possible that the diaphragm, I mean, the heart is highly oxidative, but it doesn't have a lot of satellite cells. Diaphragm is highly oxidative. Maybe it does have a lot of satellite cells. That's a knowable number. In fact, somebody probably knows it. The question is, would it be changed in CP? I'm not sure. It's a neat area, but like I said, it's not. Even though they do have a lot of respiratory therapy throughout their lifetime, it doesn't seem to be affecting activities of daily living or function. It's very interesting.
Yep.
But again, maybe it's not first line of. Maybe it's not their biggest problem.
Yeah.
Good questions, Rob. Thanks. I'll check it out.
I see, Brent, you have a question for Dr. Lieber?
I just have this incredibly naive question, which is I don't understand how sarcomeres are added in series. Do they push their way into the middle, or do they come in from the ends? That's
the dogma is they come in from the end. So if you look at the muscle tendon junction, have you ever seen a micrograph of muscle tendon junction? You got these pretty myofibrils, and then all of a sudden the myofibrils kind of split. And if you look at the actual junction between the muscle cell and the collagen of the tendon, you see this invagination, evagination, to where all of these myofibrils kind of end in a focal adhesion. And focal adhesions are common to every cell. Muscles are just, I always tell people, muscles are just really obvious with what they do. And the idea is, at least when you measure, I mean, again, these are not great experiments. They're experiments from the 70s. But when you measure incorporation of radioactive thymidine or something that represents brand new sarcomeres, you see way more incorporation at the ends of the sarcomeres than the middles. That's the dogma. But the reason I don't love it is, when you think about it, it's a weird geometrical problem. You've got this giant surface area, and the teleological explanation for that structure is by having the invagination, evagination, you increase the surface area, you decrease the stress, and therefore you make it like a stress reducer, which is kind of a neat materials idea. But the reason I don't like the incorporation idea is if you're just measuring something like incorporation of radioactivity and you've got this giant surface area, kind of course, there's going to be more there. And I don't think anyone's really done the best experiment. There is for sure myosin turnover in the middle of the muscle. You can measure incorporation of fluorescent myosins in animal muscle. And so the thick filament, which has 300 monomers or however many it has for that species, the thick filament is in equilibrium with myosin monomers, and they're always getting exchanged. I tell people, like adding sarcomeres in series is like driving your car from LA to Chicago and making it from a V6 into a V8 while you're driving. I mean, it's kind of crazy, right? The muscle's being used. And I think personally, studying sarcomere number in muscle is hard technically, so almost nobody studies it. But I think it's a really important parameter. We all teach fiber types and fiber size and muscle architecture and stuff like that. But I think serial sarcomere number and serial sarcomere number regulation is a big deal in muscle. It's just not very well studied. So I think that all those questions that you have there, some really good cell biologists could probably study it. And these days you can do it with transgenic animals, but your grant's going to have to start with, here's why it's so important. I've tried to do that already. I've said it's important in spinal cord injury. It's important in CP, stroke. I've tried to make it, make a case for normal muscle development, because that's how we learned about all the myosin genes. But it's hard to get takers for that topic.
Thank you.
Yeah.
The new sarcomeres being stuck on the end have a different myosin. They have embryonic myosin form. So you can stain for them and you can see where the new ones are simply by embryonic myosin.
Not quite. I mean, I don't think it's quite that simple. Like in development, if you look at the so-called IZI complexes and stuff like that, you start with a non-muscle myosin and then you go to embryonic and others. But if you do exactly what you say, you just take a section of a muscle at the muscle tendon junction and try to stain for embryonic myosin in a growing muscle. It's really hard to see. It's in the cytoplasm. You can measure it. But I think all that change is pretty fast. God is playing a trick on us. It'd be really nice if God gave us like a week or two to study the embryonic myosin before it goes to usually a slower isoform.
I see, Venus, you have a question for Dr. Lieber?
Yes, thanks, Rick, for a great talk. When you talked about counting the number of satellite cells in the CP muscles, so the satellite cells that were counted, are they dormant still, or they are activated? So I'm asking this because, like, if they are still, if they are dormant, this might mean that maybe they have given up on this. They're not sensing the stimulus to get activated. And so, they have even given up on regenerating the muscle.
Right. Yeah.
They are active. This means that they are trying, but they're not successful in getting like the differentiation and the full regeneration.
It's a really good question. Also, because you could imagine maybe if more of them are activated, that's evidence for this whole burnout thing. It's a really noisy system. You have to do that with multiple, it has to be immunohistochemistry. You can try sorting on, so the very first myogenic factor is MIF5, and then MERF4, and then MyoD, and you can try sorting on those things, which we've tried. It's technically almost impossible. So we did a cell sorting thing, couldn't come up with anything. That's where you get your numbers. Then when you do it in the tissue sections, so far it's pretty noisy. I'd say we see pretty much everything. It's not obvious that in CP, satellite cells are dormant, because we see plenty that show myogenic regulatory factors that are active. But we see that in normal muscle too. There is a spectrum, and I think we just are suffering right now from small sample sizes. I think it's an important question though. We have a colleague here at Scripps who's trying to do that in CP muscle, who's a world's expert in facts, and she thinks she will be able to answer the question if we can get her enough muscle.
Okay, thank you.
Yeah.
If there is no other question, Walter, did you want to give a last word?
Yeah, I just wanted to say something, you know, I've known Rick for a long time, and I know that he's always very eager to make sure that people learn from mistakes and people learn from his mistakes. And so he asked me to convey this story to you so that you can learn from things. And so he asked me to start the story and then he can finish it. He was a little bit too, you know, too humble to really start it off. And so the story goes something like that he was invited to give a keynote lecture in 2019 at a muscle conference. In fact, not only a keynote lecture, he was actually the opening lecturer for a conference. And there was nothing else going on except food and drink and him lecturing. And so, you know, we get to the lecture part and Rick goes in front of all the spectators that were, everybody's eager to go, and he opens up his computer and he wants to load up his presentation. And then he now continues the story because it's really his story, not mine.
I kind of forget. I remember things didn't work. Oh yeah, I don't think I could ever get it working, right? Is that right?
Okay, so he obviously doesn't learn from his mistakes.
Okay, sorry.
What happened is he realized that he had his wife's computer.
Oh, that's right.
Instead of his own.
He has
so
a
the moral
MacBook
of the story is never ever buy the same computer as your wife. So
that's great.
Yeah,
and let me just say, yeah, this is great. This is part of the reason I love Calgary. I always, you know, I've known of the University of Calgary. There was a fellow down here in San Diego in the very early 80s. So I've always loved the place. Between Walter and Benno, obviously they put it on the map and have done extraordinary stuff. And I've always thought that Calgary was a great place. Like, for example, you guys give a seminar and you get to have 40 minutes for discussion. Finally, right? That's what people need more of, more discussion. And you guys are great at it. And between leadership of Walter and Benno, you guys are in a great place to make the next advances. And I would say Walter and I over the years have had lots of great interactions. We don't always see things the same way, but he's a professional. He's a great guy. We've gone running together. And I'm hoping that we're going to model for the younger generation how to, you know, respectfully push the frontiers forward, because we're all trying to learn about the truth and nobody knows the truth. So thanks for inviting me to be part of it.
Thanks a lot for agreeing to do this. Always good, always enjoy what you have to say. Thank you very much.
Sure, have a great day, everybody.
Thank you.
Before we finish the seminar officially, next week we'll be hearing from Ashna Subramaniam. And the title of our talk is Effects of Shoe Upper Construction on Lateral Shuffle Movements. Thank you everyone for coming today, and I'll see you all next week.
Thanks a lot, Songwan.